Okay. Okay. We all head outside, okay? You got to try yourself. I know, I feel Hey, Amanda, how you doing? Can you hear me?
Yeah, I can hear you just fine. You look very dapper today.
What do you want us to? Man, if you're talking to me, I can't hear you.
Can you hear me now?
I can't hear you. Let me. Let's try that. Okay, Amanda, talk to me. Can you hear me?
I can hear you. Can you hear me?
I can hear you now. For some reason, let's try that again. Andrew. Okay. Okay, Andrew, can you hear me? Just turn the camera on and nod. You can hear me all right, Andrew? Okay, I'm assuming you guys can hear me.
Yeah, I can hear you.
There you are, okay. All right. Then let's try this. Andrew, you're driving, so don't do anything weird. All right, let's see. Okay. And here we go with the slide. Yeah, slide's moving.
No.
No. All right, try it again. Okay, let's try it again. Okay, slide's moving?
Yes.
Okay, good. All right. Oh, yeah. Yeah. Oh, and did you get the handout? Okay, there. Okay. Homework on the handout for today. So Zoom folks, there's a handout. I'll put the handout on Blackboard. You guys can download it.
Thank you.
Greg. That's the handout for today. This is the homework. That's due the day before we go to Thanksgiving. Okay, dokie, artichoke, time to.
Yes, sir.
Okay. The quiz has two sides. Two sides. Don't do one and then say I'm done and hand it in, then miss question five. Two sides. Do two sides. Two sides.
Got it.
Two sides. How many sides? Two. So how many sides? Yeah, I did just one side. Oh, puppy dog. Oh. Oh. Okay, for the Zoom folk, see Mrs. Schaefer about the quiz, okay? No, the one involving this, just one. No, what are the, oops. Yeah, why are they different? That should be why. I believe that's, oh, one more, okay. Now I believe that's everyone. All righty. Pyruvate enters the mitochondria. What is the fate of pyruvate once it enters the mitochondrium? That is a question, isn't it? So with glycolysis, you convert glucose to pyruvate. You make reducing equivalents in the form of NADH, and pyruvate is now going to enter the mitochondrion. Once it enters the mitochondrion, it will be converted to acetyl coenzyme A. Acetyl coenzyme A is the substrate for the Krebs cycle, which occurs in the mitochondrial matrix. Fatty acids are degraded to acetyl coenzyme A, which then enters the Krebs cycle. Amino acids are broken down to pyruvate and then acetyl coenzyme A or some other Krebs cycle intermediate. They are then fed into the Krebs cycle. So all metabolic roads kind of lead to the Krebs cycle, if you will. So the Krebs cycle is going to be pretty important for our purposes. Now, the Krebs cycle is going to completely oxidize the acetate in acetyl coenzyme A to carbon dioxide, and it's going to give 4 different reduced molecules. 3 of those will be NADH, 1 of those will be FADH2, and you'll get 1 ATP equivalent per turn of the Krebs cycle via substrate level phosphorylation. So you're going to make a bucket load of reducing agent via the Krebs cycle. What are you going to do with that bucket load of reducing agent? You're going to send it to the electron transport chain, and those electrons will go to molecular oxygen to produce water, and that movement of the electrons down the electron transport chain will be used to make a proton gradient. That'll be used to make lots and lots of ATP. That is the essential story of oxidative phosphorylation. Oxidative phosphorylation doesn't happen unless you have plenty of reductant. The Krebs cycle helps you maximize your production of reductant. So organisms use the Krebs cycle to completely oxidize pyruvate to essentially to CO2. You don't get much more oxidized than having oxidized it to CO2. So if you want to go as far as you can, essentially, with glucose, oxidize it all the way to CO2, you use the Krebs cycle. It's just that simple. And this cycle gets called the citric acid cycle because the first substrate, the first product rather, of the first reaction is citric acid. So it's called the citric acid cycle. Then it gets called the tricarboxylic acid cycle because citric acid is a tricarboxylic acid. It gets called the Krebs cycle, named after the great German biochemist Hans Krebs, who discovered it and also who discovered the urea cycle. Hans Krebs comes into lots and lots of attention when it comes to the history of biochemistry. The guy was just a force to be reckoned with. Now, the Krebs cycle, universal. You find it in E. coli. You find it in Agaricus bisporus, the Safeway mushroom that you buy at the store. Maybe it's not Safeway, maybe it's Kroger or Meyer or whatever. That mushroom uses the Krebs cycle. Baker's yeast, when they grow aerobically, they use a Krebs cycle. Those plants out there that are now dormant because it's so cold, they use the Krebs cycle. Fish use the Krebs cycle. Ferns use the Krebs cycle. Farm animals use the Krebs cycle, and so do people named Fred. The Krebs cycle is universal. And if you don't use the complete Krebs cycle, you'll often use parts of it because parts of it are so good for biosynthesis. So virtually all organisms will have some Krebs cycle organisms. Meredith, is your hand, or are you doing hand exercises? It's getting stiff, so okay. Okay. Yeah. Now, the green bacteria. The green bacteria actually use Krebs cycle to fix CO2. They run it in reverse. So they run a reductive Krebs cycle in order to fix their CO2. They don't use the Calvin-Benson cycle. Instead, they use a reductive Krebs cycle. So here we have it being co-opted for a completely different purpose. Before pyruvate enters the Krebs cycle, you have to convert it to acetyl coenzyme A. There's a step that you have to do in order to get it to that, and that's the original pyruvate oxidation once pyruvate enters the mitochondrion. So pyruvate enters the mitochondrion. It is via a pyruvate transporter that exchanges pyruvate for a proton. And because the Krebs cycle occurs in the mitochondrial matrix, you need to get pyruvate into the matrix and then ready it. Once it gets into the matrix, so the mitochondrion, the cell biological thing, not the movie, it encounters this really, really large enzyme complex. If you bust mitochondria open and you take a look at the extract of those mitochondria in the electron microscope, you will see this enzyme. It's that big. The enzyme, think upon the organism it comes from, will have a different look. In humans, it looks like a pinwheel. Remember when you went to the fair and you got those pinwheels and you blew on them? They look like one of those, except not as pretty. If you look at the PDH complex from fungi, it looks like a belt buckle. If you look at the PDH complex from plants, it looks like a flat tire. It's big, big, big enzyme. That's why it's called the pyruvate dehydrogenase complex. So this complex is also in the mitochondrial matrix. It is composed of three different enzymes. Three different enzymes. So those enzymes are pyruvate dehydrogenase, dihydrolipoyl transacetylase, dihydrolipoyl transacetylase, and then dihydrolipoyl dehydrogenase. So these get abbreviated because those don't quite roll off the tongue. So pyruvate dehydrogenase is called PDH. Dihydrolipoyl transacetylase, it's called DLAT. And then dihydrolipoyl dehydrogenase is called DLD. Also, those are the names of the genes that encode these particular polypeptides. Now, pyruvate dehydrogenase is a little different in that regard because it's two different polypeptides, an alpha beta polypeptide. And each, pyruvate dehydrogenase is actually a heterotetramer that consists of two copies alpha and two copies of each beta subunit. So the genes that encode PDH are called PDHA1 and then PDHB. There's also PDHA2 that makes an isoform of the gene that's actually specific for the Ts. And there are multiple copies of each of these within the PDH complex. So there's 30 subunits of the PDH complex of PDH. There's 60 subunits of DLAT. Then there's 12 subunits of DLD. This thing is big, folks. Really, really big. So here is the overall reaction that is catalyzed by this complex. So you take pyruvate, and you take coenzyme A, and you take NAD that's not reduced. You're going to get from this acetyl coenzyme A. Coenzyme A is going to act as a carrier to the acyl group, carbon dioxide, and reduced NAD. And the way you get there is multi-stepped and rather complicated, as I'm sure your handout indicates to you. This reaction is irreversible once it occurs. If you want to go back to pyruvate, you're going to need to go through oxaloacetate. So you need to run it through the Krebs cycle to oxaloacetate. Then from there, you can make phosphoenolpyruvate. And from phosphoenolpyruvate, you can remake pyruvate. So this reaction is irreversible. So this is an oxidative decarboxylation reaction. Oxidative decarboxylation reaction. You've gone over those yet, no, Kim? Crystal? I don't think you've done decarboxylations yet. I think that might be second semester. Okay, yeah, so look at the delta G on this. That's pretty stinking irreversible. There are 5 different coenzymes that the PDH complex requires in order to execute this reaction. Now, there's a nice handy mnemonic that you can use for this. It's tender loving care for Nancy. Tender loving care for Nancy. So thymine, pyrophosphate, flavin, adenine dinucleotide, that's the 4 in tender loving care for Nancy. Nicotinamide adenine dinucleotide, that's the Nancy part of the mnemonic. Coenzyme A, that's your care in tender loving care. And loving is lipoate or lipoic acid. If you go into health food stores, you'll find lipoic acid on the shelf. That's one of the things that they will try to coax you to buy, and the rationale behind is that it will help you with this particular reaction. That would be the rationale behind also taking thymine as well. If you don't have enough thymine, you suffer from a disease called beriberi, which is characterized by neurodegeneration. Turns out that this oxidative reaction is exceedingly important for the metabolism of neurons and the brain in general. That's why the brain feels thymine deficiency so acutely relative to tissue. So tender, loving care for Nancy. That's my mnemonic to try to get you to remember this. Okay, so here's thymine phosphate. You get it in your diet as thymine, and then it is phosphorylated by ATP, but you're going to make AMP out of that, and you're going to put a pyrophosphate group on this. Thymine, therefore, is bound by a very heavily positively charged pocket that will house the thymine. This also cannot escape from the cell at all. So this is the dietary form. This is the actual coenzyme form. Coenzyme A has that thioester, and it will use to bind the acetate that then activates the acetate for the next step, which will be the first step of the Krebs cycle. So let's look at coenzyme A. We have essentially ADP, then we have pantothene, then we have the mercaptoethylamine. Here's the business end essentially, and we're going to write our acetylcoenzyme A like this because rewriting this gets pretty tedious and ponderous. This is made from dietary pantothene. Dietary pantothene. And that dietary pantothene, if you take a look at, it's usually reinforced with pantothene. Pantothene is also found in not just dairy, but also leafy green vegetables and meat and things like that. It's lots of different dietary sources. Lipoate is a coenzyme that is usually linked to a lysine residue. This has a long aliphatic chain. That long aliphatic chain means it's going to swing from thing to another. That's very important for its function. It comes in an oxidized and a reduced form. And this is a coenzyme that is going to carry the acetyl group back and forth between various enzymes. Notice that it is going to provide a thioester. So giving this to the coenzyme A is going to be energetically a no-brainer, so to speak, energetically quite favorable. And it's linked to a lysine that's very, very common, and lipoate is used as a coenzyme. Hence the transfer. Here's your reduced lipoate. There's the sulfur. You have your thioester. So it's this is readily transferred to coenzyme A. So in bacteria, you have 24 copies of the D-lat. You have 24 copies of the PDH and 12 copies of the DLD. The numbers I gave you is for the human enzyme. You also have two regulatory proteins. You have a PDH kinase and a PDH phosphatase, because the enzyme is regulated by phosphorylation and dephosphorylation. Here is the crystal structure of the mammalian enzyme. Again, looks like a pinwheel. And yep, that's exactly what it looks like. Now, the enzymes are also named E1, E2, E3. The PDH is E1, the D-lat is E2, and the DLD is E3. So if we turn the middle light off, you don't see quite as much glare. Probably that one too. You can probably see this a bit better. Here's E1 out here, PDH. Here's E2, the D-lat here, and here's DLD, E3. So your substrate's going to go from here to here to here, from here to here to here, from here to here to here, so on and so forth, blah, blah, blah. Very complicated enzyme. All right. So 60 copies of the two polypeptides of, 30 copies of PDH, so that's 60 copies of the two polypeptides, alpha and beta, and then 30 copies of the D-lat, and then the DLD, which contains a bound. If the PDH contains a bound thymine, and then your lipoic acid is found bound to the DLAT. That would be E2. Your intermediates get passed in between this, and the intermediates are covalently attached usually. And we'll see how that works in just a bit. And to remind you, in the biochemical literature, they use this nomenclature: E1, E2, E3. E1 refers to PDH, E2 refers to DLAT, E3 refers to DLD. All right. Ready for some chemistry? Yay. Turn the lights on so you can see your handouts a little bit better. So here's the first thing that happens is your thymine has this proton extracted by a nearby glutamate, transforming the glutamate into a glutamic acid. You take this, you're going to rearrange these bonds here. By rearranging those bonds, you're then going to provide the means by which this amine is going to abstract this hydrogen here. That is going to give you this carbanion that you see here in your handout. So that carbanion, as you can see, will be negatively charged. It will now look with a lean and hungry look at this partially positively charged carbonyl carbon. You're going to get, therefore, what kind of attack will that be? It's looking for something positive, so it's nucleophilic. Nucleophilic. You get nucleophilic attack and make a bond between this carbon and the thymine pyrophosphate and the pyruvate. All right, so we formed the bond here. Now we're here. Okay, this is going to rearrange these electrons. So this CO2 here is going to take these electrons, and it's going to take its electrons and leave. What kind of a leaving group is CO2? Extremely good. Very stable, and it has this charge here, so it has the electrons to do it. So these electrons will be rearranged. You have the electron, therefore the first product that leaves is CO2. First product that leaves is CO2. Given the structure of pyruvate, that shouldn't be too surprising. So you make this enamine intermediate. That's your enamine intermediate right there. All right, so you have essentially a hydroxymethyl group. That hydroxymethyl group will get transferred to the lipoid group that is part of E2. So this here rearranges. This is your hydroxyethyl, TTP. That now gets transferred to the lipoate, and now you have a regeneration essentially of the thymine, and then you're going to abstract another carbon. You're going to have the carbanion again, and it will attack another pyruvate. So here's a transfer. Remember that lipoate is on a lysine. It swings. It'll swing up, grab it, and now you see it is bound to the lipoate. Incidentally, chemically, it's been shown this can actually jump between these two, back and forth. Yippee, yahoo, and so on and so forth, and it seems to do that. So this hydrogen can be abstracted here, then this can abstract the acetate, so this can actually bop back and forth. That doesn't affect the next set of reactions. All right, is there anything about this mechanism that's unclear? You might guess if we know it in this detail, this is a very heavily studied enzyme. It is. The literature on this enzyme literally fills a filing cabinet.
Did you post that to Blackboard yet?
Did I switch back to the Blackboard yet?
Did you post that to the Blackboard yet?
The handout?
Yeah.
No, I didn't.
Okay.
I was going to do that. So this one,
Mm-hmm.
I haven't posted that yet. I will.
Okay, thank you.
Okay. Say that again. Lipoate is right here. This is the lipoate. And here's the aliphatic chain that's linked to the lysine. Remember, that is on E2. It's on the DLAT. The DLAT. The dihydrolipoyl transacetylase. The DLAT. E2.
The acetate gets passed on to lipoate and
Yeah.
gets recycled up there back to its original form?
Yeah.
Okay.
You get the carbanion back that attacks another pyruvate. All right, so turn the page. So this simply shows you the transfer of the hydroxyethyl group to the lipoate. You see you have basic groups. Those basic groups are going to abstract the hydrogen here. When you abstract that hydrogen, you make a carbanion. That carbanion will then look very lustfully at this sulfur. The sulfur is ever so slightly positively charged. It will then attack this sulfur, transfer back to this here, and then the basic group can actually give this back to the thymine to regenerate the thymine. That's where this comes from. That particular proton, that's where that comes from. It comes from those basic groups, essentially, that you have nearby here. So you have acetate transfer to the lipoate. Yeah. So it was hydroxyethyl, and now it's just thymine pyrophosphate with this. So that's it. It was that, so it's that now. So yeah.
That isn't a free radical.
I wouldn't call it a free radical, no, because the charges are separated. Also, this is short-lived, and you give right. You also give this back to here, and then it's regenerated and ready to go again.
So which complex is lipoic acid? One, U1, 2, or 3?
Oh, E2. E2. So E2. With a little help from its basic friends. Yeah. So. The H on the, this one? That comes from this basic group here. So this one, give, and then it give us back. So this proton, interestingly, if you label these protons, they cycle back and forth between these two basic groups. The nature of those basic groups is different from enzyme to enzyme, from species to species. They're not, but the fact that they're basic is. All right, so there are actually two lipoate groups. And if you look essentially at page number 3 on your handout, it's the second page, but it's labeled page 3. The acetyl group gets transferred from the first lipoate group to the second one. So E2 actually has 2 of these lipoate groups. So in this handout here, you can see the attachment of the acetate to the first one the one the nearest to the E1, then it switches over to the one furthest from the E1. And that is the lipoate group that will give it to coenzyme A. As you can see right here, where the arrow essentially is pointing right here. So in comes acetyl-coenzyme A. This is soluble, and it will essentially attack the sulfur on the end, will attack the carbonyl, well, the thioester carbonyl, the acetylcarbonyl with the thioester carbonyl, and it will bind that there. It'll give the hydrogen that the terminal sulfhydryl group has to the lipoate and essentially produce this. You need to oxidize this if you are going to keep running pyruvate through this system. So you need to take these out, and that's what the FAD is going to do. Yeah. It's not attached covalently to anything. It's soluble. So the FAD from E3, that's attached to the DLD, the dihydrolipoyl dehydrogenase. It uses its flaving adenine dinucleotide to oxidize that lipoate back to its original oxidized state. And now you have reduced FAD, you have FADH2. That needs to be oxidized if you're going to continue to run pyruvate through it. And you have soluble NAD. That will now take those reducing equivalents. That will oxidize the FAD back to its original state. So your NAD comes in, and it will, the FAD oxidizes this, E3 oxidizes that. Now this is ready to go again. FADH2 then is going to be reoxidized here, as you can see here and here. You now make NAD plus the other hydrogen ion that goes in solution, and now you're back exactly where you started, ready to rock and roll all over again. And that's why thymine is an important part of your diet, because of the PDH mechanism. Toma? Why is the cycle important? Because if you oxidize glucose all the way to CO2, that way you make lots of ATP so that your heart can beat, so that your muscles can move, so that your neurons can fire, so that your proprioceptors tell you where your limbs are, so that your brain maintains consciousness, so that your hormones work, so that you're not supine, so that your eyes can see, so that your ears can hear, so that your digestive system works. Should I go on? No, that's why this is important.
Where do the three energies?
They're soluble.
No, I meant.
Oh, in the Krebs cycle?
Yeah.
We're getting to that. We're getting to that. Yeah, life is all about energy.
Well, I was talking specifically about the Krebs cycle.
It's important because of energy. Life depends on the constant expenditure of energy. That's why, as my grandpa used to say, that's why your mouth needs to be attached to your stomach. Well, because it oxidizes glucose all the way, so you get all the reducing equivalent you can. You milk it for as much reductant as you can get, so you can feed it to the electron transport chain to make as much energy as you possibly can. That's what it's for. Yeah, Aaron?
I was thinking, so like, this cycle mostly makes product so that it can go.
It's a catabolic pathway, yeah.
And just like my second question is where you get those things.
Yeah, yeah. This gives you the fodder for the electron transport chain. And it illustrates why thymine is so important to your diet. Without thymine, you can't oxidize pyruvate. This is very important for the metabolism of the brain. And without thymine, you suffer from beriberi. You see this in populations whose diets are rice dependent, particularly polished rice dependent. The thymine is found in the hulls. When you polish the rice, you don't have any thymine in it. And that's essentially where beriberi is seen the most. And World Health Organization and the World Bank has now given people thiamine supplements and have trained them essentially to prepare rice in ways so they could use the hulls. And so the beriberi now is disappearing from the face of the earth because of that, blessedly, because it's a pretty nasty disease. You lose the ability to move. Your behavior goes south. Your mental capacity goes south. Your consciousness goes south, and then your life goes south. Yeah. Now, excessive alcohol consumption will interfere with thiamine absorption and use. So if the alcohol consumption can cause something called Korsakoff syndrome. And Korsakoff syndrome also can be characterized by thiamine deficiencies as well. And that's part of the reason for the mental degeneration that is accompanied by alcoholism. Excuse me, alcohol abuse syndrome. Alcohol abuse syndrome. Get it right. Yes, it happens over time. So here's a list of foods that are rich in thiamine. I always get asked this question. So I put together a little list of foods that are rich in thiamine. Top of the list is, I'm sorry to say, Marmite. Yes, ooh, Marmite, you know, yeast paste, like the British eat. Vegemite, same thing. Yeah, it's gross, don't eat it. Yes, sesame seeds. Sesame seeds, like sesame bagels, very high in thiamine. Sunflower seeds, also high in thiamine. Dried herbs and spices, things like coriander, paprika, poppy seeds, mustard seeds, rosemary, thyme. Those are all rich in thiamine. Pork chops are actually rich in thiamine too. Pork chops. Pine nuts. Pine nuts. I don't know about you, but I'm a big pine nut fan. I love them. Pistachios. Oh, be still my beating heart. Pistachios. Pistachio infatuation runs in my family. I think I inherited it from my dad. Is this a Croatian thing? It is? Okay, yeah. Macadamia nuts. Quite rich in thiamine. Well, and then coming up at number 9 is fish. And then number 10 is pecans. Or if you're from the East Coast, pecans. That's the way they say. They say pecan pie, pecan, yeah. Naturally, yeah. Yeah, very rich in thiamine. It's the richest source of thiamine available. Yeast. So it's a paste made from yeast that is spread over bread and is absolutely 100% disgusting. Sounds gross, smells gross, tastes gross. And the British love it. Now on to something a bit less disgusting. Okay, Krebs cycle. So acetyl-coenzyme A, it then goes to the Krebs cycle. The Krebs cycle is a cyclic pathway. The original substrate, oxaloacetate, you're going to regenerate it so that you're ready for another acetyl-coenzyme A. Here we go, Krebs cycle. 8 steps of them are mitochondrial. 1 is associated with the mitochondrial membrane. That'll be succinate dehydrogenase. The others are in the matrix. The others are soluble. First reaction, you take oxaloacetate and you condense it with acetyl-coenzyme A to make citric acid. The enzyme is citrate synthase. Citrate synthase. citrate synthase shows induced fit, much like hexokinase and some other enzymes as well. It makes a citroyl intermediate. That citroyl intermediate contains a thioester bond. And the delta G of this, as you can see, is very, very negative. So this shows induced fit, and the binding of oxaloacetate will change. The conformation of the enzyme so that your other substrate, acetyl coenzyme A, can bind. So here is citric synthase without substrate. Here is citrate synthase with substrate. Without, with, without, with, without, with. Do you see the change of conformation? Pretty obvious. Here is the mechanism of this enzyme. Again, very heavily studied enzyme. Is that too glary, or? That's okay, turn that one off. Is that better? No? Okay, I'll leave it that way. All right, so you start out, you have this triangle of, and these are served in virtually all citrate synthases. You have a histidine, another histidine, and an aspartate. So your thioester linkage will activate these methyl hydrogens. They can be abstracted, which will occur because of this aspartate right here. So the aspartate will abstract this, and you're going to make an enol intermediate. An enol intermediate. The enol intermediate will be stabilized by this nearby histidine that will stabilize this. This double bond will now be a reactive intermediate for the attack on the carbonyl carbon right here. The enol will attack the carbonyl carbon, and then this histidine 274 is positioned to abstract the proton that had previously been donated. 320, as you can see here, will act as a general acid. It will give this proton here, making a hydroxyl group. This giving back, as you can see, is going to provide you with this here. This binding here then gives you this citroyl intermediate. That's the condensation that you have here. And then the thioester is hydrolyzed by the addition of a water. You hydrolyze the water will then break that bond, and it will create essentially the carboxylic acid here. So this oxygen goes there, and the hydrogens are then going to go to the coenzyme A. And Bob's your uncle, and there's you are. Yeah. The other hydrogen is going to go over to this histidine here. You then release this product here. That's your citrate. Second reaction is catalyzed by the enzyme aconitase. Aconitase. Aconitase is going to take the water out and put a water back in. Takes it out, puts it back in. In doing so, it rearranges this from citrate to isocitrate. This is an iron sulfur protein. It has an iron sulfur center. That's what's going to hold the molecule and allows it to dehydrate it and then put it right back in again. You can see this is not quite so favorable, but this is going to be subject to very fast reaction by the next reaction. So isocitrate will then be the substrate for isocitrate dehydrogenase. This is the first oxidation. This will create the first NADH. the first NADH, you'll also create your first CO2. There are two isozymes of isocitrate dehydrogenase. One of them is mitochondrial. It's dedicated to catabolism and the Krebs cycle. The other is cytosolic enzyme, and it's anabolic and uses NADP as its cofactor. Interestingly, this enzyme is a marker for cancer cells, and cancer cells that are show a bad prognosis have high levels of this enzyme, and it turns out inhibiting this enzyme can actually slow down, albeit not kill, certain cancer cells. So this is a gene whose protein has been very heavily studied because of its link to cancer. And the NADPH that you generate via isocitrate dehydrogenase, that is cytosolic isocitrate dehydrogenase, will be used for anabolic reactions. You lose the CO2 and you make alpha ketoglutarate. Alpha ketoglutarate. Now, look at alpha ketoglutarate. Stick an amino group, and what do you have? Glutamate. The amino acid glutamate. Exactly how you make the amino acid glutamate. Use a transaminase, you stick an amino group, then you make glutamate. One step. One step. So you can see even organisms that don't have a complete Krebs cycle have parts of it, because you need to make parts of it for anabolic purposes too. Okay, now that PDH mechanism we spent all that time on, now you have a fourth reaction that has another complex, and this complex has the exact same mechanism as the pyruvate dehydrogenase complex, and it's made up of an E2, E3. In fact, E3 that's used for the pyruvate dehydrogenase complex is also used for this reaction too, and this is the alpha ketoglutarate dehydrogenase complex, and it performs an oxidative decarboxylation, and you generate your second CO2 and your second NADH. And the product is succinyl coenzyme A. Succinyl coenzyme A. And the reaction and its enzymes and its mechanism bear a striking resistance to the PDH complex. Hence, we don't need to go over the mechanism because we already have gone over it. Just take that acetate, add 2 more carbons, and you have succinyl coenzyme A instead of acetyl coenzyme A. So we have a E1-like enzyme, we have an E2-like enzyme, and we have the same E3 that we have in the PDH complex. So we could see here, in goes acetyl coenzyme, coenzyme A, in goes an NAD, out comes an NADH, out comes a CO2, and out comes a succinyl coenzyme A after putting in an alpha ketoglutarate, and the delta G is pretty similar to that of the PDH complex. Now, ladies and gentlemen, look at what you've generated. What have you generated here? Thioester. That's a high energy bond. You can't let that go to waste, can you? So you're going to swap the energy in that thioester for a high energy phosphate, and you're going to generate not an ATP, but an energetic equivalent GTP. You're going to make a GTP. And this is succinyl coenzyme A synthetase. Synthetase. You make succinate and free coenzyme A, and a GTP. It does this through a enzyme-bound succinyl phosphate intermediate. So you make a succinyl phosphate intermediate, then you transfer that energy of that high energy phosphate to GDP to make GTP. And this is the crystal structure of the succinyl synthetase. Succinyl coenzyme A synthetase. And this delta G is kind of close to 0, so that's going to have to be pulled through by the subsequent reactions. And with this, you could see I have a histidine. That histidine will grab the phosphate. You have a succinyl coenzyme A. You make a succinyl phosphate. You transfer that to a histidine. You have a phosphohistidine. Then the histidine gives that phosphate to GDP to make GTP. And then you're across go, collected $200, you're ready to go again. Reaction number 6. Now that succinate will be oxidized to fumarate. You will not make an NADH this time, you'll make an FADH2. FADH2. This is the only enzyme of the Krebs cycle that is membrane bound. And these electrons go straight to the electron transport chain through so-called complex 2 of the electron transport chain. This is succinate dehydrogenase. Succinate dehydrogenase. So you make an FADH2, it goes straight to the ETS. And you can see this delta G is, right at 0. But you make fumarate. Now, take a look at fumarate. This is a trans product, right? You must make a trans product. This is a stereospecific reaction. The next reaction is also stereospecific because you're going to add a water across that, and that enzyme doesn't work unless you have fumarate. And we're out of time, so we will finish this. Next time, we'll talk about the regulation of the Krebs cycle, and then once we finish that, we'll get to oxidative phosphorylation and how the ATP synthase works. Have a great weekend. Stay well. Be safe. The seminar, if you're interested in it, is available, will be available online. I just need to give the link to Shaver. She'll put it online. You can listen to it. We didn't have many people there. Jenna came. It was a pretty empty auditorium, and we didn't have many people online, but I know there was lots going on at that time. So if you want to watch it, go ahead. And then if you have any questions about COVID-19, feel free to ask. Okay, folks. Have a great weekend.
Have a good one.